I'd like to introduce today's speaker. Dr. Nicholas Clark is the Associate Cooperative Extension Area Agronomic Cropping Systems and Nutrient Management Advisor with UC Agriculture and Natural Resources in Kings, Fresno, and Tulare counties. And today he's going to be speaking on insect and weed management updates in field crops. So now I'm going to pass this over to Nicholas. You can go ahead and share your slides.
And I don't like to be that person that corrects somebody. Thank you for the introduction. I'm not a doctor. I'm very grateful to have the job that I do that doesn't require a PhD, but still allows me to conduct the level of research with independence that I do. Uh, Thanks everybody for coming. I'm really happy to be presenting at such a diverse crowd from all over California and elsewhere, and of so many different professions and interests. I'm showing the title slide again here because this one includes my contact information. Anybody may reach out to me after this webinar. If there's anything you saw in this presentation you'd like to follow up on, I'd be very happy to talk with anybody. So please feel free to take this contact information down, and then I'll put it up at the very end. Really quickly, in today's webinar, I'm going to cover a few topics. I don't have a large pest management program in my research and extension work, but over the last 10 years, I've had a few significant projects, and I'd like to sort of go over and review what those problems were that we identified and how we researched solutions to them and what we found to work. So I'm going to talk about specifically issues with sugarcane aphid in forage sorghum, and then some insect problems in safflower, but in an area-wide pest management context. And then more recently, some herbicide resistance problems in small grains inside California. And then there's this image on this slide of a like a green thought bubble with a capital Q and a question mark. As Cheryl mentioned, there's going to be an exam for DPR license holders at the end of the webinar. I tried to identify the slides specifically where information is going to be pertinent to the exam questions. So whenever you see that, I guess take special note that you should expect to see information from that slide on the exam questions. I do have to acknowledge that over the 10 years that I've been working on these projects collectively, it's always been with the assistance and support and sometimes leadership from lots and lots of people, individuals within the university and outside, in like the CSU system, for example, industry, and also commodity boards, and of course a lot of private individuals like pest control advisors um, and others. The first thing I want to talk about, we're going to jump right into it, is looking at the issue with sugarcane aphid in forage sorghum. This became a problem in the United States as early as 2013, and then by 2015, sugarcane aphid sightings were being reported across the entire southern region of the United States. The belief at the time, and I don't think it's been challenged since that I'm aware of, is that these aphids, which are invasive to the United States, have blown in monsoonal winds from Central America into the southern United States, and it was in 2016 that we sort of inevitably started sighting them in California, in the Imperial Valley down by the Mexican border, and also in the San Joaquin Valley, as indicated by those red dots on the map. Thankfully, this invasive pest has a very narrow host range, but it does feed very heavily and reproduce very rapidly on sorghum and sorghum species only, as far as we know. One unfortunate coincidence is that a really common grassy weed species, Johnson grass, is also a sorghum species, and we observe it regularly on uncontrolled Johnson grass and think that it might be a supporting host for overwintering and early infestations. We find the aphid on other grasses, but it's not been documented to cause damage. And we haven't even seen it feed or reproduce on any of the broadleaf hosts that have been tested. Uh, it's a problem, especially in sorghum, because of how fast it reproduces. It it's a heavy feeder. It produces a lot of honeydew. So the graph on the right here is comparing the mortality or life expectancy, thought another way, of the aphids in the pink dots going up in that line compared to the reproductive rate or how many babies they produce. So at their median age of about 20 days, they've already lived through their entire reproductive life. Another way to put it is after about 7 days of life, then they begin their peak reproduction phase, and over the next 7 to 8 days, they'll produce on average more than 50 new aphids. What this amounts to is, population doubling between 2 and 7 days. They're a sap-sucking insect. So they reduce the amount of carbohydrates that go to the growing parts of the plants. So it can immediately cause stunting and delayed development. And for sorghum, which is a grain crop, it can look like diminished grain production or no grain production at all. Also, the honeydew, which is a natural secretion, from the aphid, gets deposited on the leaves of the plants, and it supports sooty mold growth, which is not a direct infection of the plant, but it has a black or gray opaque appearance and blocks sunlight from the leaves and reduces photosynthesis further, which is also a problem that can limit growth and yield in the crop. In this picture in the bottom left, the plants to me, they appear very dry. This is a pretty heavily infested field. As you can see, a lot of the live aphids and some of their molting on the bottom of the leaf there, and then even on the panicle or the flower stalk. It's a very heavy infestation, and they've been known to destroy entire fields when they're not checked or controlled. Early on, we tried to do some work to quantify the impact of um, aphid presence. So how many aphids are there in the field and how long are they on the plants? We tried to relate that to yield, and we did see a little bit of a relationship where the more aphids you have and the longer they're on the plants, you see a reduction in yield. But for any statisticians, you might be looking at how tightly these dots cluster around the line, which is not very tightly, or you might be looking at this R squared value, which is relatively low. It's a poor relationship. We weren't able to find a strong relationship in the small data sets that we had. But I have some slides later that show some indications that aphid feeding definitely does reduce yield, at least in our trials. Uh, Jennifer Heggie, one of our dairy advisors in the northern San Joaquin Valley, and I did some work monitoring sorghum fields in 2016 on 16 dairies, and we looked at the feed uh, quality profiles for select constituents like dry matter, protein, fiber, ash, so on and so forth. And we compared what do those values look like between aphid infested versus non-infested fields. So not to say anything about how many aphids were there or for how long, but what were they present or not. 6 of the fields did not have any aphid present at the time of harvest, and 10 of them did. And these little stars above the bars here indicate that there was a statistically significant difference in these feed constituents. So, for example, we saw crude protein increase slightly when there was aphid, Acid detergent fiber went up. That's the fiber that's more difficult to digest or less digestible for the animals. Typically, we like to see that lower in a higher quality feed. Ash went up. That's not good. That's definitely non-digestible, and it's um, just minerals that aren't necessarily helpful for the animal diet. Starch went down when there was aphids, and that's whole plant starch, which was expected because we saw much less grain development, which is where most of the starch accumulation is in the sorghum plant. And starch is an important feed constituent because that's one of the main drivers of milk production in dairy cows. And then non-fibrous carbohydrate, which would include starches and other simple sugars, went down, which is expected because it's very highly related to the amount of grain produced. So we did see early indications of strong impacts on feed quality with aphid infestation. We didn't do any work in California that added new information to how to scout an ID for this. We tried to respond very rapidly to the problem, and we relied really heavily on the southern states that had this aphid for about 3 years before we did. So the next information is derived from what they had to say. So first thing is being able to distinguish this insect. They can be from a light green to a vivid orange. That's quite a range of colors. They've got black antennae. They have these black tailpipes, otherwise called cornicles. This is the sugarcane aphid on the left, and this is a more typical green bug aphid, which is also found in sorghum. They've got black feet, but so does the green bug there. They're very, very heavy honeydew producers. So anyone who's familiar with aphids, other honeydew producers might be like whiteflies, for example. It's that shiny substance that you find on leaves. I think I have a picture coming up here where I might show that to you. So here, there's not a lot of distinguishing features between the sugarcane aphid and the green bug, except these tailpipes are pretty distinct. They're short and black on the sugarcane aphid, and then they're longer and pale colored on the green bug. There was a lot of confusion about what we were seeing when it first came to California, caused a lot of problems. There was also some confusion when some yellow type cotton aphids were appearing in cotton, and it really scared a lot of growers who are in the same region as these sorghum growers, and sometimes the same growers themselves would grow sorghum and cotton were finding this cotton aphid in their cotton, and the similarities are kind of striking when you're looking at those black tailpipes there. They thought they had sugarcane aphid, but the Eugene Hannon, the ag commissioner entomologist at County of Fresno, put out a really cool bulletin that you're seeing here to help with the distinction and identity between these two. These tailpipes, or the cornicles on the cotton aphid, are fairly long. They're about the same length as the cotta, or the short little tail on the back side of the aphid here, whereas on the sugarcane aphid, they're very short and stout. Okay, so that was a big distinguishing factor. And then just reaffirming that the sugarcane aphid, as we know, does not feed or reproduce on cotton. But one big distinguishing factor, again, hint, all right, big question here, is that these cornicles, or these tailpipes here, are short and black on the sugarcane aphid. Here's just a really nice close-up image that Dennis Haynes from the Kings County Ag Commissioner provided for us on a sample that we brought in of a large nymph sugarcane aphid. And then here are some more juvenile nymphs next to actually a winged adult. Just a cool close-up picture there for you. How to scout. This is what we recommend. So first, look at the edges of the field weekly until the pest is identified. That's typically where they show up first. They're going to pop up on the field, on the edges of the field. Once they're there, then look at 4 representative areas, say like 4 corners of a field, no larger than a 40-acre management block at a time, staying 50 feet away from the edge of the field or a road. And in each of these sampling zones, selecting between 15 and 20 plants, and then collecting a top and a bottom leaf, and then counting the aphids on them. And the treatment threshold is whenever the average number of aphids per leaf on 25% of the plants reaches 50 or above, it's time to treat with an insecticide. What does 50? Oh, let me show you here. Here's a good picture of what the plants look like on the edge of the field when that infestation first starts to come in. You can see on the left picture a lot of the molting of the nymph aphids collecting on the top side of the leaves. And then in the picture on the right, the live aphid nymphs prefer to hang out on the underside of leaves, collecting there. And then you can see this sort of shiny, wet look on some of the lower leaves as the honeydew sort of mists down and collects on them. And then there's even a wet soil appearance here. That's actually honeydew. So, I mean, they're, like I said, they produce a lot. It's a very easy to identify visually sign that the aphids are present on the edge of the field. This is what 50 aphids look like. This is obviously a leaf that's very heavily infested, but this small circle here, there's approximately 50 aphid nymphs in there. Another way to think of it is an average sized adult pinky, well-trimmed pinking nail. That about that area filled up with aphids represents 50. So it doesn't take a lot of aphids to trigger a treatment that's economically beneficial. Again, that question mark up there, 50 aphids per leaf is the treatment threshold that other state extension agencies um, put forward and we support. So we did some work on trying to define how we would control this pest in California. We know there are some preventative control measures just based on what we know about the biology and from work from other states. Manage Johnson grass, no alternative hosts. So get rid of those alternative hosts. There are some resistant varieties. Those need to be chosen with assistance from seed providers and maybe the sorghum checkoff program also has some information about varieties that are showing resistance to sugarcane aphid. We have very, very limited information in California about the relative tolerance or susceptibility to sugarcane aphid between varieties. We just simply haven't done that work, but we do have some data. Planting early and encouraging early vigor helps because the aphid tends to show up a little bit later in the summer growing season. And as this table on the right from Mississippi research shows, comparing in infestation timing, so pre-boot or before the head is emerged versus when it's just emerging at boot, panicle emergence or the flower stalk after it's emerged up to soft dough, which is the grain heads have actually start to develop and the grains themselves, the starches are starting to dry out in there and actually resemble the sort of bread dough. Um, when they're infested at 20%, 20% of the plants are showing an infestation and they're not controlled, we see up to total yield loss when that's happening as early as pre-boot, and then as little as 20% yield loss when that happens at a later stage. So planting early can help avoid the more susceptible stages interacting with aphid infestation. Um, but also there are some neonicotinoids that are available as seed treatments, not for purchasing and applying on their own, but the seed manufacturers and suppliers will be able to provide those products, and they provide up to 40 days of very good protection against the aphid infestation after crop emergence, and we've seen good success with that. At the time we were doing this work and promoting that, I think about 50% of the growers were adopting that practice and seeing good results. And then what to do when you have the aphid, when it shows up. So a more responsive method. Um, the insecticides that we have available that we've seen work are flupyridifurone or Sivanto or aphidopyropen, Safina, applied as a foliar treatment with enough water for good coverage. Sorghum forages tend to have very dense canopies. Coverage can be an issue. Savanto is a systemic product, but it only travels through the xylem, so it's only going to move up through the plant with the transpiration or the water pattern. So efficacy or control against the insect is only going to be as good as that product gets deep into the canopy. Um, Green bug poison is sugarcane aphid energy drink. So malathion, dimethoate, and then chlorpyrifos, no one has to worry about in California. It's not registered anymore. But these were the typical broad spectrum insecticides that were effective against the green bug, and they were showing no control of sugarcane aphid. They were just lapping it up and continuing to reproduce happily. We had a trial, a replicated trial at the Shafter Research Station down in Kern County. This graph is showing the number of aphids per leaf on the y-axis or the vertical axis. And this red dotted line going across the graph is just marking that sort of 50 aphid per leaf, just to give a reference line for what our treatment threshold is. And then each of these colored lines, zigzagging across the graph, are showing the level of aphids between the different treatments. So each line is a different insecticide treatment. And as you move from left to right on the graph, we're seeing time progress forward. And the key down here is when it says 6 DAT, that means 6 days after treatment. So any number of days and then days after treatment. And the only products that we saw provide consistent, acceptable levels of control from the beginning of the trial at 6 days after treatment to nearly 40 days after treatment were the Savanto Prime and the well, at both rates of 4 and 7 fluid ounces per acre. Transform or sulfoxaflor, which we had a lot of hope for, didn't look too great under this high pressure early in the season. And none of the other insecticides that were more traditional provided good control. The next year, under much, much lower aphid pressure, with the same insecticides, Transform, which is shown by the line with a little X marker on it, did perform better. Dimethoate, which is shown as this white or open square, didn't perform well. We were pretty consistently above what we would have called a treatment threshold. Um, but we just had much lower pressure that year, and we were actually seeing a lot more beneficial insects coming into the plots, and we hypothesized that might have helped some of these treatments along as well. We were really interested in this sort of new experimental deal with using Savanto. Uh, Other states were applying it in the soil because it is mobile in the xylem or the water conducting tissue in the plant. So if the idea was that the plant can take it up, it would move throughout the plant and provide residual or long-lasting systemic control. In other words, it would be effective against insects that were feeding on parts of the plants that we didn't apply it to. It's taken up through the roots, moves throughout the rest of the plant, and then provides insecticidal control when they feed on the leaves later. So we tested applying Savanto at planting and then at lay-by, or this sort of side dress application, the last opportunity for making a side dress application in the field. At planting, we applied it on the seed in the seed furrow before closing the wheel, like as a drench. And then at lay-by, we mix it with UAN32 fertilizer and did an offset application below the surface. And in this graph here, we're showing same thing, aphids per leaf on the y-axis, and then these different treatments. The IF means in furrow or at planting at different rates, 4 and 5 fluid ounces, and then FOL, foliar, is when we applied it to the leaves like a normal, aerial or ground application. SD for side dress, meaning we applied it at the same time we did fertilizer. Then this DAI down here on the bottom axis means days after introduction. We actually brought aphids into the field from a commercial field to sort of seed it around our plots there. And then when we got when our plots that didn't have Savanto already applied in the soil got above that 50 aphid per leaf, we treated with the foliar. And the treatments where we applied Savanto with the seed had control over the aphid for more than a month after planting. They provided excellent control. Um, as you can see here with these lines down on the bottom below the red dotted line, and the side dress application, these two square markers here, their treatment efficacy broke about 20, somewhere before 21 days after the foliar treatment, but they still provided better control than doing nothing at all, which is this open circle here. This is what our yield estimations or measurements looked like. I put this green line across to sort of make a level reference with the yield we got with the untreated control, which is about 22 tons to the acre. So we saw, a yield protection on average of about 1 to 5 tons per acre by using the Savanto treatment in there for all of these. But the variability in these plots was so high we couldn't find any statistically significant differences, but the average difference is very clear. We tested the feed quality, and one thing that we noticed in these same plots is the acid detergent fiber was significantly lower when we used a foliar treatment. So, 34.8% acid detergent fiber in the foliar treatment or the in-furrow or at planting treatment. The neutral detergent fiber was significantly lower when we made the foliar treatments and the in-furrow treatments, and the starch was significantly higher when we used that as well as compared to the uncontrolled. So it did have a positive impact on feed quality. What came of it is aphidopyropin or safina has since been registered in California for use on sorghum. Transform has had lots of troubles in getting registered and can't even get a Section 18 because of a lot of pollinator issues and failure to demonstrate economic benefit of having it. And then Savanto, fluparitifuron, was registered as a foliar, but also has since been registered for that soil applied use. So there's a lot of options with that product. Now moving on to talking about the safflower insecticides project really rapidly here. Safflower in the region that I'm familiar with in the southern San Joaquin Valley is almost exclusively grown in the Tulare Lake Basin and around the actual geographic feature or geological feature of the ancient lake itself, the lake bottom and the lake edge at approximately 30,000 acres per year, and it's very, very commonly grown in a tight rotation with processing tomatoes and cotton. Um, it has a lot of benefits, as safflower does. It's grown for oil that can be food grade or processing grade, but it's a good soil conditioner. And in the lake bottom area, there are going to be perched water tables with poor water quality that's not conducive to cash crop growth, like with tomatoes and cotton. And we can see that safflower roots actually draw up poor quality water from as deep as approximately 9 feet, so they can lower perched water tables. Um, but they also are big insect havens. The safflower fields are. There's a lot of lygus, a lot of stink bug, a lot of beet leaf hopper. So they sort of provide this early warning system where safflower fields are monitored for these pests early in the season when tomato and is just being transplanted or when cotton seedlings are just coming up or just starting to square and provide an indication for when those pests might be growing to a population that's important for those cash crops. There's a lot of products that are registered for use in safflower for that state activity on lygus, stink bug, and leaf hopper. There's one, two, three, four, currently 5 known modes of action represented among there, but I'll say that 100% to 99% given the year of the modes of action used fall within these top 4. The cycloniliprole or the diamide is brand new, and I think it's maybe applied on probably like a couple hundred acres this year commercially. Um, and most of these products are going after lygus, where very few of them are going after leafhopper or stink bug. In on the label for safflower for these different products, this is just a quick listing of what the max pounds of active ingredient per acre per year are, and sometimes that translates to sometimes they have explicit statements about the max number of applications. Sometimes there's just a max amount of AI that can be applied, and then we can kind of infer that, well, if someone's applying the top label, like with dimethoate, for example, they're going to hit the max allowed in just one application, or they can split it up and get fewer. But there can be quite a few number of applications going after these pests in safflower over the year, more than 10. And that might sound like a lot, but just a couple years ago when the lake bottom flooded for the last time, it was followed by a very, very heavy lygus year where a lot of the farmers in the area were reporting needing to apply insecticides to control lygus up to as many as 13 times in the same field. So we did some work with support from the IR4 project, which looks at pest management solutions for specialty crops in the United States. I'm going to go over 4 modes of actions that are new to safflower in the United States and especially in California: cycloniliprole, acetamiprid, aphidopyropin, and isocycloserum. And then our target insects in these trials were lygus, beet leaf hopper, and green stink bug. And then just a quick reminder, we mentioned that the safflower is a good crop in this rotation system because it provides a good monitoring site for an area-wide IPM program where growers can get an understanding of the population dynamics of these pests that are really important in cotton and tomato, not so much in safflower. That's not to say that they're not important in safflower at all. It's just that it's very different dynamic. Lygus, for example, the treatment threshold in cotton, depending on the age of the cotton, can range from like 1 up to maybe 10 counts per 50 sweeps, whereas in safflower, the treatment is somewhere closer to like 40 lygus per sweep before it becomes damaging. So it's hardly thought of as a problem in safflower. But these other insects like beet leaf hopper, for example, pose a lot of problems to tomato because of vectoring of virus, and green stink bug can cause problems in both tomato and cotton. So that's really why we're interested in looking for solutions in safflower to prevent the spread of these insects to those other crops. I'm going to go over just a few select results, but a quick explanation. We compare all of these to what we call a grower standard treatment, but depending on the year and the availability of insecticides and the design of the experiment, the grower standard treatments varied quite a bit between these different products and mixes. And then all the treatments, when called for, were mixed with appropriate adjuvants like surfactants and oils and or fertilizers and defoaming agents whenever they were needed. So we're going to look at cycloniliprole in 2019. What you're looking at you'll see on the left the name of the insect and the growth stage that's shown on the graph, and then very similar to the early graphs you saw, we're seeing days after treatment. On the graphs where you see these vertical green lines, these indicate when we actually made treatments or retreatments. And then anytime you see like a red bracket, or in some cases a red arrow, indicates when there was statistical significance or a mathematical difference in treatment control of the target insect. So with the lygus nymphs, for example, it wasn't until after the second and the third treatment where we were actually seeing cycloniliprole do better than the untreated control, and it was usually for a very short period of time after those treatments, and it didn't appear to. I don't think they ever got as good as the grower standard. This is the same treatments, but looking at the total ligus. So counting what was the previous one? Mean ligus count. Oh, this is the nymphs, and then this is the adults. Excuse me, sorry. And there were only a couple of days toward the tail end of the experiment where we were seeing some differences, but there's quite a lot of variability there. This is just one year that I'm showing. In 2020, we were looking at acetamiprid. Same graph, but looking at Lygus nymphs, we were only able to find 1 day after the second treatment, about 5 days after treatment, where we were seeing acetamiprid perform better than the untreated control. When we were looking at the adults, there were a couple days after the first treatment where it was performing as well as the grower standard and better than the untreated control, but that trend fell off for the rest of the experiment. That only occurred up to about 6 days after treatment, the first treatment. With beet leafhopper, on the other hand, we were able to see a lot more control. So, up to 6 days after treatment, 6 or 7 days after treatment, we saw very good control of beet leafhopper after the first treatment. And then after the second and third treatment, we saw similar trends. Even though the counts were low, the data were very clear that this was acetamiprid was effective at controlling beet leafhopper for about a week after treatment. Notice the green question mark up there in the top right. This was an interesting one. Another question mark up there, so to pay attention to, is false chinch bug just blew up in the acetamiprid treatments. Notice the scale here. We were counting several thousand of them. This is a big deal, not necessarily in safflower, but really important because this early season migration of a heavy population of false chinch can actually kill cotton seedlings, tomato seedlings, and young tomato transplants. So we weren't happy to see that, and we were very surprised to see it actually. Okay, 2023 aphid, also known as Safina in some circles. In this trial, we were mixing aphid with novaluron. That's what the registrant or the chemical manufacturer wanted to see in the IR4 trial. We also looked at a high rate of aphid for crop safety, and I included those data in here. But looking at Lygus nymphs, when mixed with novaluron, we were able to see good control of Lygus nymphs with that treatment throughout the entire experiment period. So it looked good, it looked like a good material. Always better than the untreated control, and a lot of times similar to the grower standard. Looking at the same graph for Lygus adults, the trend was very, very similar. Good control of the adults throughout the period with that mix of aphid and novaluron. Here's what the beet leaf hoppers look like. Numbers weren't as promising. There were a couple of dates where we were able to see the aphid and novaluron mix doing better than not doing anything, better than untreated control, but didn't present any consistent trend. And the numbers were often so low, the number of insects were so low that it's hard to determine what that actually means and make any conclusive statements about how well this product controls beet leafhopper and safflower. Here's some isocycloserum. It's a brand new AI. I hope I'm not misspeaking. I don't think it's registered anywhere in the United States for anything yet, or if it is, it's just happened. Strongly effective at controlling Lygus adults. As you can see here, always better than the untreated control, almost always similar to the grower standard. And that trend is the same for the Lygus nymphs. It does appear to actually be a better product on the nymphs than it does the adults, which is not uncommon with Lygus. Most products have a hard time going after the adults themselves. Here's what it looked like on leaf hoppers, similar to with the aphid mix. We didn't really see a lot of differences in treatments except for a couple of select dates. And again, the counts of the leaf hoppers were so low, numerically speaking, that it was difficult to decipher what these data meant. So we didn't make any strong conclusions about how well this product controls beet leaf hopper in safflower. So where did this leave us to date? Cycloniliprole, it's been registered as Harvanta 50 SL, but for suppression of Lygus adults, remember, it didn't really, have that knockout power that the grower standard did. It would just kind of bring it down a little bit below what the untreated control did. And growers reported that that was satisfactory for them because remember, it's one product that's in a toolbox of other products that they're using to rotate different AIs in their in those fields to control this insect to try and prevent it from moving into cotton and tomatoes and having to expend their insecticide applications in those crops. Acetamiprid, as of February of this year, that package has been submitted to the US EPA or Environmental Protection Agency for review, which is the normal process to try and get that product registered. So my understanding from people who know this better than I do is that we're still looking at best case scenario a couple years out until it's something that may be registered for use in safflower in California. After it goes to US EPA, California Department of Pesticide Regulations takes another crack at a evaluation packet before it can be registered for use in California. And then for the aphidopyropin and the isocycloserum, the last a few graphs that I showed, we're hopefully in our last year of field evaluation. We've done all of the treatments. We're just counting all the insects and doing the analysis. It looks very good. They both look very good. The manufacturers and the IR4 program representatives are have unofficially stated that they're happy with the way the data looks. We don't anticipate there being any problems with respect to proving that these are effective products. But there's much more to it than that than when registering. So best case scenario, maybe 5 years down the line, we might see these in safflower. Um, Winter forage weed management issues. These are some common weeds that we find in small grain forages in California, a mix of grasses and broadleaves. They're all problems for different reasons. I mistakenly left wild oat off here as an important grass, but should not be neglected. It's definitely a large problem. Um, what we were seeing a few years ago, what PCAs were telling us is that they were seeing common chickweed that they used to be able to control with pyroxylum and tribenuron or Simplicity and Express herbicide in California. It wasn't controlling it anymore. So what we did is we put those very patient PCAs through the ringer. We went out to the field and just asked them the litany of questions about, what could have gone wrong with the application that might explain why they weren't seeing it control the weeds as they expected to. And there's lots of things like with the environmental conditions, right? Was it controlling other weeds? What about areas that weren't getting sprayed? Does it look similar to the area that you did spray? We went through their application records. We even went to one of the fields and tried to kill it ourselves with these herbicides. And when we couldn't, we took seeds to a shade house of the weeds and tried to control them later. And I'll show you results of that. This is what the PCAs were seeing, and this is a treated field, and this is what I observed with them is at tillering or a young stage. You can see the treated chickweed. I'm sorry about the resolution here, but these light green dots, that's the chickweed. About three weeks after it was treated, not controlled. Same field as the wheat or triticale is extending or growing taller. You can see the chickweed growing up there in the canopy. with the crop, and then here's it flowering when we're starting to initiate grain development. You can see the chickweed grown up right there with the flag leaf. This is a really big problem because we anticipate that this would affect yield and feed quality. So we asked, well, are ALS inhibitors, which pyroxylum and tribenuron are they a problem? Do we see a lot of herbicide resistance? And we looked to a global database and found, well, yeah, one of the most commonly occurring instances of herbicide resistance occurs with ALS inhibitors. Um, we asked ourselves, well, might we be helping this problem along in California? And we looked at application records of different herbicides applied in small grains across the state in the DPR database. And we found, well, of all of the different modes of actions between ALS inhibitors and synthetic auxins, more than 60% of the treated acres are actually being treated with ALS inhibitors and synthetic auxins, where ALS inhibitors are the leading number there. Um, so we were applying a lot of ALS inhibitors, and that could potentially can be contributing to pressure for developing some herbicide resistance. We said, well, what if we start searching for cases of herbicide resistance in chickweed in some of these databases? What does that look like? Well, what we found is, if we're looking at North America, um, it was almost always found in wheat, or a small grain, sometimes in alfalfa, sometimes in peas, and it was always exclusively an ALS inhibitor. In California, just spoiler alert, eventually what we found is, mesosulfuron or osprey, pyroxylum or simplicity, and then tribenuron or express. We did document that there is herbicide resistance to these AIs in some chickweed that we find in small grain cereals in California. This is us putting out a trial trying to control the chickweed that we found in one of these fields where they weren't able to control it. This is what happened afterwards. You're moving more to the right is good control. And it wasn't until we did 4 times the label rate of the express that we were able actually to get something that looked better than the untreated control. But there did appear to be something that looked kind of like a dose rate response. So the express, and we went 2 times the label rate. We were able to do a little bit better than the untreated control, but it didn't separate. It wasn't significantly different. With the simplicity or the pyroxylin, even up at 4 times the label rate, we didn't get control. So we took seeds of the weeds from these infested fields. We sprayed them in the greenhouse, or sprayed them and then put them in the greenhouse and let them grow. And I'm going to show you the results of those. I'm going to go through these quickly. At the top, you'll see the active ingredient and the level of treatment. This is untreated control. And then each row of these plants of chickweed are weeds from a different population, so from a different field. The bottom row here is from an organic field that had no history of being treated with ALS inhibitors, so we expect these herbicides to kill it. Okay. This is what happens after 4 weeks when we sprayed with pyroxylum at the label rate. We killed the organic control, but couldn't kill any of the suspected resistant ones. And that's the same ingredient, but at 8 times the label rate. So we want to see these dead on the bottom because they're susceptible, and we'd like to see these dead, but we didn't kill them, even at 8 times the label rate. Here's tribenuron, same arrangement of populations there. There's at 1 time the label rate. There's at 8 times the label rate. So we did see a lot of stunting, but there still was some survival, as indicated by that green tissue there, even at 8 times the label rate. Here's mesosulfuron or osprey. At 1 time the label rate killed all the organic stuff, didn't kill any of the others. And at 8 times the label rate, they're thriving. Imazethapyr, which is actually registered in alfalfa, untreated, 1 times the label rate, and then 8 times the label rate. We actually saw some differences between the different populations there. So probably not as widespread of a resistance to that AI. Here's with the Mazamox, also registered in alfalfa, untreated, 1 times the label rate, and then 8 times the label rate. Yield is affected in fields where the triticale was infested with weeds. We saw as much as a 37% reduction in yield compared to the weed-free areas. There's some other major issues in small grains like uncontrolled ryegrass and uncontrolled shepherd's purse. I don't think we're running out of time, and I don't think that any of this other material is in the exam, and I hope to maybe answer like 1 or 2 questions. So can we open it up to at least a question if there are any?
There were a few questions that came in. So here's one. Why is the sugarcane aphid called that if it only grows on sorghum?
Yeah, excuse me. So that was a mischaracterization on my part. The sugarcane aphid also does affect sugarcane, but we're I was referring to crops that we commonly grow in our region. So my apologies.
All right. And then there is one in the chat. For your Savanto Prime trial, did you try a combo of both in-furrow and side dress?
No, we didn't look at a combo of in-furrow and side dress, but the industry was conceiving of these different application modes as um, ways to sort of address potential infestation at different times of crop growth. So, for later plantings, we were encouraging like an in-furrow at time of planting application if they wanted that systemic control because it would provide control for more than a month at a time when we expected aphid infestation to be more likely. And we said with those side dress treatments, for earlier plantings, it provided a little bit of leeway. They could kind of wait and see, what's happening in the neighborhood. Are aphids popping up? And if they had time when they might be doing a side dress application with some fertilizer, they could apply then. So no, we didn't do them at the same time.
All right. And then I'll just ask the one last question. This question is, did you guys do any work with tank mixing or chemistry rotation for weed management?
We did we didn't do any tank mixing or chemical rotation experiments for weed management for the chickweed in small grains. Um, I did have the privilege of looking at quite a number of industry trials out in grower fields, and we saw that one for the ALS inhibitor resistant chickweed in small grains, we saw that one sort of promising tank mix was a very early timed treatment with good coverage of clarity or dicamba and ET or pyrofluoroethyl. So one is a synthetic auxin, the dicamba, and the other is a PPO inhibitor. The combination of those two did a very good job at stunting, burning up and stunting the chickweed for long enough for the crop canopy to close in and essentially choke the weed out. Um, we did a little bit of work that I didn't show, looking at some different adjuvants, so like MSOs or modified seed oils versus non-ionic surfactants with Express, because we saw that even in the resistant populations there was a dose rate response. So we thought, well, maybe if we can make the mix a little bit hotter, somehow we can still get better control, even though there's some resistance genes in this population. And we saw that I believe it was using a modified seed oil did provide a little bit of a hotter mix and control the chickweed more, but there are some cautions there with respect to crop safety. So working with the PCA on that is paramount because that is a hot mix that could ding up the crop a little bit, especially depending on any other chemicals in the mix, like a PPO, like Shark, for example.
All right. Thank you. And so thank you so much, Nicholas, for your presentation today. We appreciate you being here.
Okay, no, thank you all. It was a pleasure.
All right. So I hope everybody enjoys the rest of the afternoon, and we'll hope to see you at the next webinar. Have a great day, everybody.